So, my name is Michelangelo Campanella, and I'm going to talk to you about the role of the mitochondria in the regulations of cholesterol metabolism and relevance in the signaling associated with the cell survival. So, I obviously presenting my own data, the data generated with my team, with some supporting slides from other colleagues, and obviously all this reflects my opinion. And goes with that I have to disclose no financial conflict of interest, and especially in the material I'm presented. Obviously, I'm incredibly sorry by that, but hopefully with time this will change and I have a more important disclosure to make. So we are very much focused on the mitochondria. This is what we do. We are the mitochondria cell biology and pharmacology unit, and what we do is simple and hopefully of interest for today's audience, which is the uncompromised functional characterizations and pharmacological validations of mitochondrial mechanism. The reason why I'm here, and I'm very grateful to be here, and grateful, of course, to the organizer to bring in all us in this wonderful site, is because I've been given the challenge to try and take you through the world of mitochondria with our latest advancement on the subject. So, potentially, some of you knows about the mitochondria, but it's by far the most important organelle within mammalian cells, conserved also across other eukaryotes. And the story of mitochondria seems to be the following, that in ancestral eukaryotic cells were colonized by this organelle or small organism itself of a prokaryotic origin. And together with the host cells and what was then the primordial mitochondria, they establish a very good endosymbiosis. I think we can all agree on that. And potentially the reason why mammals are the dominant animal on the planet is because they have this fantastic ability to couple oxygen into energy, providing obviously a tangible evolutionary advantage. But mitochondria have retained their independence despite being in the same environment of the host cells. And this independence, as you all know, is well translated into the different DNA that bear with us. But what is interesting is that over the last 25, 30 years, we learned that beyond the core ability of the mitochondria to generate energy with unique efficiency, they are quite pivotal in the regulation of cell signaling and, most importantly, of the demise of the cell. So, in other words, the mitochondria are very cooperative organelles, but as soon as they are crossed, they become quite selfish and independent. And so in order to preserve their essential bioenergetic competence, because this is what they do every milliseconds of their life, they are ready to drive the cell to death. And obviously, with greater knowledge on physiology and physiopathology and the know-how that we develop in several diseases, the attentions on mitochondria burst. And particularly so when we discussed the topic of this summit, which is the aging. So the correlation between mitochondrial aging is very strong, and indeed aged tissues are characterized by, let's say, corrupted mitochondria. So here I share with you a snapshot of electromicrographs of mitochondria from young tissue, young muscle, which we can call an healthy mitochondria. You may appreciate the fantastic ultrastructural morphology of this organelle with those cristae where respirations occurs, and on the left-hand side of the screen. And on the right-hand side, instead, you can see a unhealthy mitochondria that belonging to an old muscle. This is a screenshot that I got from the eminent Professor Zorzano in Spain. But this is well indicate that defective mitochondria do underline age-related conditions and aging of tissue. Obviously, analysis on that account have been several, and indeed more and more the model has aging as a mitochondrial disease really took space. And aspects that have been quite rightly so mentioned at the round table at the end of the previous sessions, that were about commenting on the proteostasis relevance, the stress of misfolded protein. This is basically what happens with the mitochondria. So an healthy genotype, you have a functional organelle with all the proteostasis in place. And instead, when this equilibrium is lost, obviously mutations occur, and the mitochondria per se, but particularly the environment surrounding the mitochondria, so the hosting cells, is altered. And indeed, what is the differences between physiology and pathology and healthy tissues, diseased tissues in respect of mitochondria, is a fully functional organelle or a dysfunctional organelle. And the big question is that are we accumulating dysfunctional mitochondria as we age? And unfortunately, the answer is yes. But so our main goal for our research is really to understand what drive and regulate this transition, because mitochondria are numerous within the cells, and they operate as a network communicating one another. But obviously, if something goes wrong, the communications is disrupted, and the mitochondria do not respond well to stress, any kind of stress you can think of, the transitions from healthy to pathological mitochondria occurs. So, and particularly, whilst trying to understand what make this organelle transitioning from healthy to unhealthy, we were curious to see how the challenged mitochondria adapt to stress and respond to stress. And if you think this has been quite a recurrent theme in these 2 days, the ability to adapt from a dietary kind of insults or lifestyle or conditions, and the way we or the mammalian body respond to these adaptations underlines the outcome towards longevity or aging conditions. And so, and when we started to study this, obviously we had to come up with a target that it may be a reductionistic kind of approach, but obviously from a molecular and pharmacological point of view, the only one that we could pursue at that time. And we started from the pharmacological angle, and in the literature there were evidence of a specific molecule on the mitochondria, which was a binding site for one of the most sold medicine on earth, which are the benzodiazepine. And this specific binding site, called initially peripheral benzodiazepine receptor, was able to bind the benzodiazepine that did not outside the central nervous system. And with time, we learned that this molecule binds not just exogenous ligand, but also endogenous one. So this protein that initially was called peripheral benzodiazepine receptors changes denominations in translocator protein because this protein does not only bind to benzodiazepine, but bind with quite an important endogenous element, and this element is cholesterol, as you can see here. So obviously, since the discovery of this protein at the end of the 70s, the attention has been immense on it, and there are several chemical regulators. Some of those are also available to treat patients. And despite this exploited pharmacology, if you want, and attention in the development of this kind of tool, the role, the physiopathological relevance of this protein was not been clear. And so what the most recent bulk of literature indicated that this protein sitting on the mitochondria, it is a mitochondrial stress response protein associated with cellular pathology. So that really for us was a good way forward to try and inform the transition in between healthy and pathological organelle. And indeed, as I said, the attention on the chemical that bind this protein have developed a series of tools enrolled not just for a therapeutic outcome, but also for a biomarking and diagnostic outcomes, such as here the images I'm showing of the PET analysis of inflamed cell. So a protein that define a unhealthy mitochondria, a protein which allow us to score the unhealthy mitochondria, and to do so not just in vitro but also in vivo, might have a relevance for age-related conditions, and this is what I will show in my talk. Among those conditions, our attention focused on the Parkinsonian type of disorders, because colleagues in Manchester showed us that patients could be classified in their progressions of the disease according to the degree of overexpressions of this tiny mitochondrial protein. And so we studied this. And in order to inform the biology behind this clinical data, we obviously looked more carefully to the mitochondria. And what we saw that if we force the expressions of the protein by means of molecular biology, we not only alter the structure of the organelle, which you can see is looking much more dense, more packed within a standard mammalian cells. This is HeLa cells which are overexpressing the protein, and a fluorescent molecule, GFP, that allow us to see the mitochondrial network. But most importantly, according to the increase in the amount of organelle within the cells, also the redox stress, which is, according to everyone, a prodromic parameter for cell pathology, was incremented. And the very opposite did occur if we in the lab ablated the protein. So a protein that is used to score the presence of Parkinsonian disorders is also a protein that leads to redox stress in the cells. And so the conclusions was that indeed the mitochondrial homeostasis is very much modified according to the level of TSPO. However, the cells are equipped with a very sound homeostatic mechanism which remove the faulty mitochondria. This process is called mitochondrial autophagy or mitophagy. And what we discovered was that the overexpression of TSPO was able to counteract this mechanism of quality control. So everything really looked to make sense. You have a protein which is used to score the severity of a condition that is related to the mitochondria, and what does this protein do? This protein blocks the quality control of the organelle, thereby sustaining the developing of a pathological onset. And obviously, this was well recapitulated at neuronal level in human-derived neurons, in which the overexpressions of the protein lead to a loss of mitochondrial quality. But more interesting for us was to see that not just the molecular regulations of the protein led to the modifications of this mitochondrial network quality, but also a series of toxins that are associated to sporadic form of Parkinson were able themselves to trigger the expressions of the protein, indicating that the upregulations of TSPO did work as a maladaptive parameter for the cells that is exploited in pathology to generate or and unveil all the more serious onset. Whilst we were studying this and happy of our conclusions, we saw that the repressions of the stress-induced mitophagy via TSPO had an impact at a transcriptomic level. We're now trying to understand whether this is of genomic or epigenomic nature or both, but that was the connections with it. If we corrupt the mitochondria, or the cells themselves start corrupting the mitochondria by responding to a stressor via the upregulations of the SPO, we have a mechanism by which modulating the mitochondria, we modulate well the core of the cells. That even though I care very much of mitochondria, I must admit that in mammalian cells, the nucleus too has is importance by expressing genes. And so we started to study this mechanism of mammalian cells adaptation to stress, which is called the mitochondrial retrograde response. And so we then devised a theory by which the increment of pathological mitochondria had an impact on the nucleus by the exploitations of this pathway. This was a concept shared, not just shared by several labs worldwide, but the understanding of the mechanism and the obtainment of secure readout and so target to intervene of this adaptive mechanism were really scarce, at least in mammals. And our theory, our working hypothesis was if this empowered mitochondrial retrograde response, which underlines every progressive pathological onset, was enhanced by the proximity of the mitochondria with the nucleus. So you have to think of a cell with its organelle within, and if you increase the amount of mitochondria that defeat the control mechanism, where are these mitochondria going to go? Everywhere. And so our idea was, is the proximity of the nucleus important to expedite this pathological alteration? And so, and we worked on that using several cell models, and I'm happy to see that we did so in comparative fashions using human murine and companion animals derived cells. And what we were able to recapitulate is that chemical or physical stressor produce a redistribution of the mitochondria to the nucleus. This associates with the overexpressions of our target molecule, TSPO. And this overexpression leads to a proximity of the mitochondria onto the nucleus. So this may seem just a trivial observations, but thankfully it turned out to be not the case. Indeed, the proximity between two organelles means greater ability to communicate, a target to try and alter the process that by that proximity are depending. And particularly so in the case of cells is the need to produce a subset of genes to withstand stressor. So if the mitochondria change their locations and go to the nucleus, they are able to convey a series of messages that allow the cells to adapt to the stress. But what if these interactions is overexploited, overused? What is going to be the outcome? Certainly the outcome is going to be pathological, and this is what I'm going to convince of. So these nucleus-associated mitochondria, 3 years ago, following our publications, hijacked the attentions of all the community interested in organelle physiopathology and cells adaptations to organelle alterations. And these specific molecules that establish interactions are also conserved among species, and since then there is a economy. And the conservation of this point of contact is very relevant to really advance our comprehension on the progressions of conditions or the adaptations to those conditions. So, and so far, what do we know about these functions and why the point of contacts are relevant in mammalian cells? And in order to try and investigate the relevance of these interactions, we focused on another age-related disease, and particularly cancer. Why is that? Because the comparative oncology is a growing field, and also one of those that we are more interested in. So we're trying to establish a parallel between human cancer and companion animals one. And still with the goal to inform the transitioning from healthy to pathological mitochondria and how these impact the metabolism of the cells. So if you recall my initial slide, I tried to explain you that this protein, which is very susceptible to environmental changes, let's call it like that, besides being overexpressed, has got a specific daily job. And the specific daily job is to bind the cholesterol, which circulate from the cells, and put it in the mitochondria to fuel the metabolism of cholesterol, and particularly the formations of pregnenolone and all these steroids precursors. So when was the time to think of a cancer model to study, we focused our effort on the mammary carcinoma, because it's renowned to be an hormone-driven disease. And obviously, the initial analysis, which was also confirmed by other labs, did show that the overexpressions of this protein positively associates with the aggressiveness of breast cancer. And along with that goes a specific accumulations in the nucleus of all the pro-oncogenes that you want to screen. So, what was nonetheless particularly fascinating for us was that these formations of interactions, consequent overexpressions of the SPO, had also another fascinating role in the cells, which was the redistribution of cholesterol from the mitochondrial to the nucleus. So that was incredibly fascinating, because even though we thought that there was a cholesterol-related mechanism in this, let's say, unhealthy relationship between the mitochondrial and nucleus, we could never imagine that the accumulations of cholesterol on the mitochondria, which could not be metabolized, was then given to the nucleus. So, And this was indeed the case, and the image, the cyanine blue staining of the cholesterol, hopefully is quite convincing to show you how the portion of the mitochondria are deprived of these lipids to give it to the nucleus. And which was the goal of this? The goal of this was to drive the cellular resistance to endocrine therapy. And at the very moment cholesterol was depleted by the cells by pushing the metabolism of cholesterol by the mitochondria, or by using a drug that we all know, the statins, you completely block this pathological mechanism of evasion of endocrine therapy. So here we are trying to highlight that the ability of the mitochondria to bind and traffic cholesterol does end up into a pathological outcome unless this is proper metabolized. And in support of our findings, there came a retrospective study in the Netherlands. We show that breast cancer patients that are undergoing standard of care with endocrine therapy, they maintain the efficacy of those treatments if at the same time they undergo statin treatment. And so this was actually quite an important observations for us, because really help us to provide in vivo patients relevant evidence for our findings that if the cholesterol in excess, which is present in the breast cancer cells, is used in incorrect fashion, it ends up working as a proto-oncogene signaling route. And of course, we show that by pharmacologically regulating the NAM, still using the statins as a control, we were able really to increase the efficacy, not just of endocrine therapy, but of other tools that defeat the growth of breast cancer. So this finding, which is certainly important from a fundamental aspect of cellular adaptations to stress, and also on the need of managing the lipids level in order to avoid severity of the conditions or evasions of endocrine therapy. And this is not trivial things, because the evidence that obese subject, they develop breast cancer more than the other is there, as well as the issue that are presented when it's come to the standard of care, has now a cellular explanation. But what was the relevance for animal biology of this and the medicine that is trying to address the aging-related disease? So, as you all know, and especially this audience know, companion animals do develop cancer. And when they do this is particularly violent and not always treatable. So our effort was to try and see which were the basis for these fast pathological evolutions, and also the impossibility in most of the cases to use therapeutic protocols based on pharmacological agents. There is a serious issue related to toxicity. So our hypothesis or aspirations was, if we find a way by which we can improve the efficacy of endocrine therapy, we can hopefully try and devise protocol which require far less of these toxic agents for animal. And obviously we went down the route to see if the molecular mechanism of adaptation driven by cholesterol were recapitulated in companion animals. So to this end, we used again the canine mammary carcinoma, and we appreciated that as for human cells, our mitochondrial target, TSPO, does present a positive correlations with aggressiveness of the disease. And this is now being seen by various means. Here I'm just showing some histopathological analysis. But obviously, in order to investigate the underlying mechanism, we had to use a model that allows us. So from tissue-derived patient, we moved to cell lines. And thankfully, there are quite reliable canine mammary cancer cells line that mimic the stage of disease, and the analysis of such a model was important for us to see that indeed there is a positive correlation between the accumulations of TSPO and the severity of the disease. But which was the metabolic relevance of that, and did TSPO work in the same way as it does in the human counterpart? So in order to do though, first of all, we checked whether there was a difference in susceptibility to endocrine therapy. Here I'm showing data with tamoxifen, in which we see that obviously the estrogen positive cells do respond to the treatment. Estrogen positive cells which present a lower level of TSPO, which is on the other hand sky-roofed in the most aggressive counterpart, the REM134, which instead do not respond to tamoxifen. And exactly as we saw for the human cells, if we are able to ablate this pathway that retrocommunicates to the nucleus to force the expressions of oncogenes, we are able to obtain a susceptibility to the treatment, but particularly not anymore relocalizations of the oncogene-specific transcription factor. But what was interesting for us was to see that all this was related to the ability of canine mitochondria to handle cholesterol. So cells that present susceptibility to the treatment have a lower level of mitochondrial cholesterol, which indicates greater metabolism by the organelle, and so the pushing of the pregnenolone pathway. If instead TSPO, and therefore the binder of cholesterol, is increased, the mitochondria cannot metabolize cholesterol efficiently anymore, and so they jam the system, and by the increased level of the metabolism that you can see here in this brighter cyanine level, represent a mechanism of defense against the endocrine therapy treatment. So we try to see this in a more sophisticated model that is thankfully now open to the canine-derived cells, and we appreciated that also the 3D reconstruction, the 3D growing of the cells was particularly susceptible to the agents that target this cholesterol pathway. And now the effort we are having is to devise the protocol which allow the use of reduced amount of endocrine therapy to deliver efficacy on canine mammary cancer cell associated with the targeting of this cholesterol pathway. So we proved that this reprogramming can be obtained in vitro. So we cultured the less aggressive form of canine mammary cancer cells for 14 months in estrogen deprivations or exposed to tamoxifen. And guess what? We had a specific upregulation of this mitochondrial target, TSPO. So that all makes sense, but data, and this is the last one I'm showing to this audience, was potentially the most interesting because thanking our collaborations with veterinary colleagues from Zurich, we access patients' data. And by checking the level of TSPO in the transcriptome that they provided us, was particularly fascinating to see that following neutering, which is a procedure that is adopted to prevent the outbreak of breast cancer, guess what gets immediately upregulated? TSPO. So neutered animals are somehow pre-primed to the outbreak of this pathology. And one of the explanations we are given is that this is why when the form of mammary cancers occur, you have those to be happening quite severe in these patients, potentially because there has been a metabolic reprogramming that led to this prodromic priming. So, and this takes me to the conclusions that I really would like you to take with you for the rest of the afternoon and ideally for the evening and beyond, is that stress mitochondria respond well by establishing point of contact with the nucleus. This is a mechanism to escape cell death, but is a mechanism of maladaptation. And so think about Parkinson's disease, that all of a sudden outbreak in age subject without having any indications before or diagnostic. So that this now favors the redistributions of cholesterol, whose metabolism is key to the expressions of pro-survival gene, that this is a conserved mechanism, and so a conserved targeted mechanism both in human and companion animal. And the most important point, maybe not conclusion, but point of discussions is that these pro-pathological conduits that depends on the mishandling of cholesterol does offer new opportunity for the management of patients that can go beyond pharmaceutical and hopefully including nutraceutical. With this, I would like to thank all my team that has been working on that with me for 4 years, and obviously the funder, particularly the European Research Council and the PET plan that let us obtain the core informations I've shown you today. Thank you very much.